Okay, so what I wanted to actually do was to look at enzymes, their structure and their function. So we know from GCSE and from the A level that we've done that enzymes are proteins, and we now know that proteins are sequences of amino acids that are folded into a secondary structure and then folded into a tertiary structure, which is held by hydrogen ionic bonds and disulfide bridges. So how does an enzyme actually work? Well, the first model, which we also learned at GCSE, is the lock and key model. It's the idea that the enzyme has a specific tertiary structure, a specific shape, and within that, the active site has a particular shape, and that shape is complementary to a specific substrate. The substrate fits in to form an enzyme substrate complex. The bonds get distorted by the enzyme, and therefore the reaction happens. What you've done is you've lowered the activation energy because enzymes work as a biological catalyst. They change the rate of the reaction, but they don't get used up themselves. Now, it's really important that when we are describing enzyme reactions that we refer to specific terms as listed here. So tertiary structure instead of shape, active site, complementary, specific, substrate, and enzyme substrate complexes. So if I was describing how the lock and key actually worked, this is how I would describe it. I would say an enzyme has a tertiary structure so that it has a specifically shaped active site. You can see straight away that we are using terms that we listed up there: tertiary structure, active site. This is complementary to the specific substrate, and they bind to form an enzyme substrate complex. And the distortion of the bonds in the substrate causes a lowering of activation energy, leading to the reaction. So when you construct your answer for an enzyme question, make sure that those terms are included. The second model is the induced fit model. The induced fit model is slightly different because what it does is it says that the active site is not the actual complementary shape to the enzyme. In fact, it's a slightly distorted shape. And when the active site fits into the substrate, all it will do is that the active site will actually mold to fit the shape of the actual substrate. So whereas in the lock and key it's fixed, in the induced fit it actually can mold. So what are the factors affecting enzymes? We've come across these before: temperature, pH, enzyme and substrate concentration, and inhibitors. And we'll look at each of these in turn. So if we start with temperature, this should be a reaction graph that you are used to. Temperature against rate. As you increase the temperature, if we put some numbers in, say from 0 to 40, the rate increases, and then above 40, as you increase it, the rate decreases dramatically, and the peak is called the optimum. So the question is, what is actually happening here and here when you're asked to describe the process? So when you're asked to describe it over here, you must refer to kinetic energy. So as the temperature increases, the substrate and the enzyme have more kinetic energy and move faster. Because they're moving faster, there are more frequent collisions and therefore more frequent enzyme substrate complexes are formed. So what about over here? Well, once you go beyond the optimum, we know that the enzymes denature. Now note, they don't all denature at once, which is why you don't get a vertical drop, but they do denature over a short period of time. So what does it mean when the enzymes denature? Now remember those words we were talking about earlier: tertiary structure, active site, complementary, specific, substrate, ES complexes. We've got to put all those words into our answer. So what do we say? We say that the hydrogen stroke ionic bonds break, and therefore the tertiary structure and active site change. This means that the substrate is no longer complementary and cannot bind. No ES complexes are formed when all the enzymes are denatured. Note that if only some of the enzymes are denatured, you'll get less enzyme substrate complexes forming. Okay, so what about pH? So again, this is a graph you should be used to. We've got two enzymes here. They both have their optimum pH at slightly different pHs. So this is a pH 2, so it could be the enzyme found in the stomach, for example, pepsin. This has a pH optimum about just under 8, so it could be something like amylase. So what do we say is happening either side over here? So it's exactly the same principle as we were looking at before. The ionic bonds break, the tertiary structure and active site change shape, specific substrate is no longer complementary and cannot bind. Less or sometimes no ES complexes are formed. So it's fairly straightforward. You'll note again, I'm using all of those keywords that we mentioned before. Okay, so what about enzyme concentration? So enzyme concentration is slightly different. You'll see the concentration of the enzyme increases, the rate increases. Now, it might increase forever, but it is possible that it will level off. Now, it's fairly obvious why it's increasing here. You're getting increased concentration, so you're going to get more frequent collisions, which leads you to more enzyme substrate complexes per second. But why might it level off? Well, the only reason it will level off, and it doesn't often happen, is if the substrate has become limiting. In other words, we just don't have enough substrate for the reaction to go any faster. You get a similar graph when you do substrate concentration. And you measure the rate. And the beginning will be the same. So you've got more frequent collisions, more enzyme substrate complexes forming. You are much more likely in this for the enzyme to be limiting. And what's actually happening here is that all of the active sites are full. And so even if you add in more substrate, as all of the active sites are being used at once, no increased rate can happen. It will just stay at a constant rate all of the time. Okay, so that leads us to the inhibitors. Now, inhibitors form one of two types. They can be competitive or non-competitive. So with competitive inhibitors, the inhibitor has a similar, note I've underlined similar, shape to the substrate, and therefore it competes for the active site. Now, the important thing about competitive inhibitors is that they are reversible, because basically the inhibitor pops in and pops out again. Now, this is important because it means if I look at a graph of substrate concentration against rate, with an inhibitor, even though the rate is lower than without it, as you increase the inhibitor, as you increase the substrate concentration, the rate will increase. Okay, and that's because the more substrates you've got competing with the inhibitor, the more chances are that the substrate will get into the active site instead of the inhibitor. Another graph, another way to present it is to measure the volume of the product against time. So for example, if hydrogen peroxide is broken down into water and oxygen, and we look at no inhibitor, or we look at the presence of an inhibitor, obviously with no inhibitor, the reaction increases, then levels off because you've actually used up all of your substrate. It's gone, so the reaction's over. This is different to the other graph. We're looking at volume here, i.e. how much is actually produced. But with an inhibitor, it's all going to be slower, but you're still going to get eventually to the same amount of product because you had the same concentration of substrate to begin with. A non-competitive inhibitor is an inhibitor which binds to what they call the allosteric site, which is a place away from the active site. It changes the tertiary structure, and therefore the active site changes shape and is no longer complementary. So once again, we can think about using those words here. Okay, if we were trying to describe it, we would say a non-competitive inhibitor binds to the allosteric site and changes the tertiary structure and active site. Therefore, a specific substrate is not complementary and cannot bind, reducing the number of ES complexes. It could be reduced down to 0 depending on the amount of inhibitor you have. Again, notice I think about those using those keywords every time I write an answer. So with the same graph as before, so volume of gas product against time, as you increase the time, obviously the reaction is happening until the point at which the reaction stops. But with a non-competitive inhibitor, it will basically increase a little bit, and then once you've denatured all of your enzymes, it will stop. No reaction will take place. Please be really careful at looking carefully what's on your graph. Okay, so there are two graphs that we've come across here. Okay, one is volume, okay, and the other one is rate. Okay, so be really careful there. Okay, so with a non-competitive inhibitor, the substrate concentration will have no effect on the rate at all. It is independent of the rate and it is irreversible. Now, I want to just show you those two graphs again, okay? Because it's really easy to get them mixed up, okay? So this is why I say you've got to be really careful about what you put, what you're looking at. So if we had, for example, the first one where we had time against volume, you end up with a graph that looks like this. It looks on the face of it very similar to this graph because the shape is almost the same. But the reasons are completely different. Here, we are looking at the process of one reaction, and the reaction volume of the product increases as the reaction takes place. So this was the reaction before. In other words, you are measuring the volume of oxygen produced, and it will go up and up and up, and then it will stop being produced. It will level off, i.e. no more volume. And that's because you've run out of hydrogen peroxide. This reaction, what we're doing is we're changing the concentration of hydrogen peroxide and measuring how quickly a certain amount of oxygen is produced. And as you increase the substrate concentration, the rate gets faster to produce that certain volume until the rate is constant. So you're still making oxygen, but you're making it at the same rate each time. And that's when the enzyme has become limiting. So just be careful on these two graphs that you look really carefully at what is on the two axes.